Hello, everyone. Well, welcome to the Drug Hunter Flash Talks today. I'm your host, Dennis Koester, and I'm the Director of Industry Research and Relations here at Drug Hunter. I'm joined today by our CEO, Dennis Hu, and by Ashwini Ghogare, who will talk to us about generative AI in drug discovery. We're super excited to have you here as a guest today, Ashwini. Welcome.
Thank you.
Really excited about your talk, Ashwini. I know you and I met in the past to talk about generative AI and this interesting tool that MilliporeSigma is building. I know in the community, there's a lot of excitement about AI and really curiosity, right? I think we keep seeing news about AI-based companies getting funded, taking programs to the clinic. We have a couple articles on this on the website, but I think people are dying to know when are they going to be able to actually use it themselves? What is this stuff? And is it coming for our jobs, or is it going to be a useful tool to help us? So thank you for making the time to join us today.
Absolutely. The pleasure is all mine. I think it'll be a good discussion across the potential of AI, but also the challenges. I think we all want to be able to use it, but we have to be careful to make sure that we have many other considerations before in terms of data and others, which we'll talk about today.
Ashwini generously agreed on having audience questions throughout the entire Flash Talk. So if you have any questions during the presentation, during our interview, please put them in the Q&A box. You find this in the bottom of your Zoom bar as well. And yeah, so without further ado, I wanted to introduce our guest, Ashwini. So Ashwini is the head of AI and automation for drug discovery in the Global Science and Technology Group at Merck KGaA in Darmstadt, Germany. And there she is responsible for setting up the AI-powered drug discovery and automation portfolio. Ashwini is a leader in the AI field and has extensive experience in software development, new business incubation, and also software product sales. Ashwini spearheaded strategic initiatives to identify transformative AI technologies for drug discovery applications. And she and her team successfully built and launched new AI-powered drug discovery solutions. And the one that we're going to be talking about today is Edison. And we'll, again, hear more about this throughout the discussion. She also is extensively involved with MilliporeSigma's flagship retrosynthesis tool that's called Cynthia, and we'll talk about this today as well. Ashwini holds a Ph.D. in organic chemistry, and her graduate work actually led to the creation of a startup, which is called Singlet Oxygen Therapeutics, and that startup is focused on photochemical drug delivery systems. Ashwini, that's a really impressive background, and we're really excited to have you here with us today. So thank you for making the time, and we're super excited to talk to you today.
Awesome. Thank you for the kind introduction, and I'm equally excited to talk to you and the audience here.
Awesome. Awesome. Yeah, before we get into the topic of generative AI, we like to ask some icebreaker questions. So I wanted to ask you, what inspired you to start your career in drug discovery?
Yeah. Thank you for the question. I think this is a question close to every drug hunter's heart. I think we all sort of embark on this journey to ensure that we get better therapies for patients much faster. Somebody in our family, somebody so close, has usually gone through disease areas that's been challenging in finding cures much faster. And so if us as scientists can contribute, even a small drop in a big ocean, it still makes a big difference. And so that's been my motivation. My motivation has been to build these therapies and leverage technology to be able to do that.
Awesome. Yeah, I can very much relate to that myself too. Yeah, I myself suffered from a disease called Bell's palsy. Yeah, so I'm definitely relating to that as well. This is my motivation as well. We share that. Cool. So today, what are you excited most about in drug discovery? Is there any technology, any particular platform that you? I mean, I guess I know your answer, but just asking.
Yeah. So I mean, I feel multiple technologies that are exciting in this era. I know we're going to focus on generative AI today, But automation is, I think, another technology that I'm super passionate about. Generative AI, in my opinion, is a tool that can help, and we'll talk a bit more about this on pattern recognition and cool insights, but the data behind that is super critical. And so how can we make automation or leverage automation to create these data sets that are going to be important for the training of these models? To me, that is another area that we can kind of continue to innovate around.
Yeah, no, absolutely, absolutely. Cool. So one question that I always find fascinating is if you could go back in your career to the start of your career and give yourself some career advice, what would that be?
Oh, well, let me think about it. Well, if I were to go back and tell my younger self, I would say the same that I told, the new generation of scientists. I wish I had gone back and spent more time in data science. I feel as one, I kind of always refer to this new generation as the trilingual or bilingual generation that has experience in drug discovery, that are scientific experts, but also have deep understanding of data and algorithms. And that trifecta is crucial, especially when they are contributing towards scientific algorithms and scientific discoveries. So I would definitely ask my younger self to spend more time on data science and sort of close the loop there.
Yeah, no, that's a great advice, I think, for people today, and I think that's what we're kind of looking for. What should people today do in their careers to be successful? Yeah, so I guess before we go into the generative AI topic, I wanted to ask you, of course, what you like most about Drug Hunter.
Wow. So Drug Hunter, in my opinion, this is one, I would say, publication, magazine, website, however you call yourself, most read and most visited by medicinal chemists, whether it is scientist level all the way to director and the head of chemistry. You have been able to bring together up-to-date literature on what data is published, what molecules are published, clinical trials, acquisition. I mean, there's a whole breadth of information that is important for medicinal chemists and drug discovery scientists to stay on top of. My entire team always keeps an eye on this and continues to learn a lot. So thank you for the remarkable effort your team has put together and continues to put together for all the scientists.
Awesome. Yeah, no, that's great to hear. We create this product to be used by others, and it's amazing to hear that it's helped your team and that your team is following the literature. Yeah, awesome. Thank you so much. All right, cool. Let's get started with the generative AI topic. So as you know, and as you mentioned, we have a lot of medicinal chemists in the audience today. So could you explain to them generative AI, in drug discovery, maybe in more general terms to, unveil the curtain on what that actually is?
Absolutely. Let's start with the general term. I think generative AI, as everybody knows, is an algorithm that allows you to create or that generates new content, whether it's natural language, it's music, right? It could be new patterns, but it's all based on the recognition of patterns and the data it's been trained on. Now, if we take this in for medicinal chemists specifically, it's a powerful tool to be able to generate new molecules based on the properties that you're looking for, to be able to explore larger chemical space, which are humanly difficult, and to be able to generate insight, which are challenging or unrecognizable, let's call it, through larger data set. And so generative AI has helped or is helping, I would say, significantly when it comes to new drug discoveries, determining synthesis feasibility, as well as new molecule discovery overall.
Cool. Yeah. So, and if you were to highlight the strength of generative AI that maybe, generative AI could do better than a human chemist, what would those strengths be?
Yeah, so to answer that question, maybe we could jump to a slide
Yeah.
that
Awesome.
would help to describe that really well. And maybe before we jump into that, let me start sharing my screen. And we want to start with a disclaimer. So let's first figure out screen share. All right, let me know when you see it.
Yeah, I can see it now. Perfect.
Perfect. So as part of this organization, before we share any content, I want to start with a disclaimer that MilliporeSigma is a business of Merck KGaA, Darmstadt, Germany. And we're known as EMD Serono, MilliporeSigma in EMD Electronics in US and Canada. So if you hear different names, that's all us in the same organization. So with that out of our way, to answer your question in terms of the power of generative AI or where can it really help, I think a couple of strengths of generative AI are able to look at huge amount of data and recognize patterns out of it that can help us get better insights. If you look at the larger chemical space, right, and here's an example. If you look at the space, even with the newest technologies, we have been able to only explore anywhere from 10 to the 4th to 10 to the 9th chemical space, which may feel significant, but as a medicinal chemist, I think it is still quite small. And this is a space where you have drug candidates that are approved or chemical spaces that are available from many vendors. But if you step back a bit further and maybe some more, you will see that there's a huge amount of unexplored chemical space that could be leveraged to find the undruggable, to drug the undruggable sites, as well as to find novel compounds that are important for drug discovery. And so how can a human, right, or be able to explore such a large chemical space? And this is where the power of generative AI really comes into play, to be able to do this fast, compute, and especially that it's compute intensive, and to be able to identify recognition patterns that can give us new insights in whether it's new molecules, the properties of these molecules, and then tying them back into drug design and synthesis.
Awesome. Yeah, no, that's right. That I mean, I've experienced this myself as well. Like, yeah, looking at large data sets, AI is definitely superior. So, that maybe help us a little bit understanding why we don't have to fear for all of our jobs now. So tell us a little bit about the weaknesses of generative AI today that you're seeing.
Yeah, I mean, I'll share an example. This sort of question had come to me when I started very early in this career. And I still remember I was in a room with a lot of scientists and I was talking about Cynthia. At that time, Cynthia was, a very new tool. And a whole room of, audience, I was asked this question. So is it here to replace me? And that was a chemist. And absolutely not, right? I think all these tools, and I still remember that day, it was a very humbling experience. But I wanted to highlight that personal experience because I'm a scientist, you're a scientist, and all of us are, I think, incredible at generating and designing new molecules. But I think there's a huge space where these generative models can help us and empower us. And that's where Cynthia helped, and that's where generative models such as Addis and others could help as well. And if you look into specific challenges, right, while generative AI has multiple strengths we talked about, but there are many places where it can really add value or where we have to watch out for to drive it. It's the large chemical spaces, exploring them while taking properties into consideration, as well as synthesizability. If you step back for one moment and just think any generative AI that can generate language can generate molecules. At the end, it is a string of smiles or entry keys. But the problem is that all of this, the molecules that it'll generate are normally garbage if they don't have context for drug design. And that's where the accurate property prediction is super, super important. It's important to govern the generative AI and guide it to say, hey, look for chemicals in this property space. And the other area where it has challenges and would use guidance from scientists is in synthesizability. Because at the end of the day, if you cannot make these compounds, then whether it's generative AI or it's human, it's not really going to be valuable to medicinal chemists.
Totally, totally. Yeah, you got to be able to make those compounds too. You want to put compounds in a vial, is what many of my managers said to me before. So yeah, you mentioned this commercial tool that MilliporeSigma has that's called Edison. Can you tell us a little bit more about what that is and maybe why a medicinal chemist should use it?
Absolutely. So, and again, we'll go through a couple of slides and then we'll come back to the flash, conversations as well. I wanted to show you a little bit about Addison itself. So as you looked at the challenges and the promise of AI, this is where we started asking question, can we build a tool based on the methods that are known and used today, and then bring the generative AI models to complement it, right? And the goal is to really be able to empower our scientists to be able to do drug design faster. And so we focused on 3 different areas for Addison. The first one that you're seeing here is to be able to explore these vast chemical spaces. There are 2 kinds of spaces, right? You have the known chemical space as well as the unknown chemical space. The known chemical space is relatively, there are various methods to be able to search it, but the unknown chemical space, that's where generative AI is super helpful. And that's how we deployed the reinvent models in the back end to expand into these large chemical spaces. But beyond the chemical spaces, there are 2 other parameters or opportunities that we saw. And these help in addressing the med chemist day-to-day workflow, right? And so what is med chemist really trying to do? They're trying to find compounds by virtual screening or other methods. They're looking for ability to do scaffold hopping. They're looking to do fragment design and decoration, and they're looking to do optimization of molecules as they go through different workflows. And this is where we strengthen Addison and utilized the core of Addison, which is trained on 30+ years of known pharma data. And we'll talk a bit more about this, but that was important to have robust experimental data that can drive all these workflows. And at the same time, we have the Cynthia integration as well, which allows to drive synthesis know-how, whether you call it synthesis accessibility of these molecules, or to find retrosynthesis pathways for the molecules that you want to make at the end. And like you said, you want to be able to make it in a bottle, right? That's the end goal for a medicinal chemist. And so this was our essentially North Star, let's call it that way, is to be able to put together ADME predictions, retrosynthesis, and allow medicinal chemists in their day-to-day workflow to explore chemical spaces, do lead ID optimization, and take it forward in their projects.
No, that's super cool. And yeah, you already kind of hinted towards like that the data is important. So could you maybe share with us how you were able to get access to those training data, these high quality training data that was needed for Addison?
Yeah, absolutely. So as we started looking into building Addison, given there multiple different solutions and new generative models coming into play, we very quickly realized that these models were generating compounds that were not accurate when we started testing them, testing them. The amount of make and test time required was way more than design. If you see a medchem workflow, it takes about 2 to 3 months to complete a design, make, test analysis cycle.
Mm-hmm.
Imagine you were investing as a med chemist this much amount of time per cycle, and then these molecules fail, right? That's essentially the biggest challenge for med chemists. And so in order to ensure that you have success through this process, it's important that the molecules you generate in step 1 have higher predictability, right? Have higher robustness and higher let's call it success or probability of success. And that's where we recognize that it's the data actually that is causing us problems. So we started with our healthcare groups in Merck KGaA, Darmstadt, Germany. So just for context for the audience here, Merck KGaA group has 3 different functions. We have electronics division, we have life sciences, and we have healthcare. So our healthcare, which is focused on oncology, immuno-oncology areas, they have been generating experimental data like any other pharma for many years. And they spend incredible amount of time in last 5 to 8 years in cleaning that data and distilling it down to the data points that were assayed the same way. And I say this specifically because the fundamental problem in combining data sets is that people, or the flaw is that people tend to combine data points even when they are done different, or the execution or experimental execution has been done differently. And that's the real problem. You can't really compare apple and oranges, even though they're both fruits, to put it simply. And so it was very important that we distilled down the data and brought it to clean data sets that have been assayed similarly, that have different broader chemistry groups to it, and train our models on that data. And so this is just an illustration to show you how we train the models in the back end. We had to reiterate the models as we generated, we trained the models, we got predictions, right? We took the predictions to the lab, we synthesized them, and we brought that data back to retrain the model to ensure that they are driving the right direction. And so a lot of, I would say a big thank you to our collaboration with our pharma colleagues in the space. They have been our champions throughout this process.
Awesome. Yeah. And, now medicinal chemists, I mean, it seems like they help providing the data, which is awesome. But, in general, medicinal chemists, as I can personally attest, are like a critical type of folks. So maybe can you talk a little bit about the challenges that you had in getting medicinal chemists to use this day to day?
Yeah, it's quite interesting that you get the data from the same folks, and then still there's skepticism. But that is not surprising. We're all scientists, right? We're all skeptical of our own data and everybody else's.
Right.
Which is to be just a bit of segue here, which is part of the issue why there is no publication of negative data. So before I go on the trust and confidence, I think this is one of the fundamental gaps, is there's a lot of positive data in this space, and there isn't any negative data for the models to be trained unless they have been in-house developed and unless you have these thousands of data points done experimentally. And so that was, I think, one of the areas why we chose to train it on our internal data. But as we started rolling out this with the medicinal chemist, one of the important asks for any medicinal chemist in the area is to ensure that the predictions are as reliable as possible. And the reliability of these predictions come from the confidence level of these. And so here, what I'm on the slide here, if you look on the left-hand side, this is the distribution of data. And this is, we're taking an example of a KGaA permeability model. The distribution of data, you can see there's a combination of permeable compounds, non-permeable, and partially permeable compounds. And so the question for medicinal chemists is, that they ask us is, this is a great distribution model or distribution data, but how do I gain confidence in this? And so we kind of worked through this and added another parameter called confidence score. So as you saw from the previous distribution to now, right, the second distribution is essentially saying, what is the confidence level of this model in each of its prediction? And so what you can see here, if you look at the third column, it's predicting it has 93% confidence that which of the compounds are going to be permeable, which is a very small fraction. And the confidence score of 0.6 is a good estimation of saying that this is either permeable or not permeable. And so adding that confidence score to your distribution data helps to narrow down and ensure that each prediction has strong confidence around it.
Yeah, that makes a lot of sense. So you know essentially when to trust the data and when you have to be a little bit more critical as you design your compound having the right properties, then you can feel better about it. If the model is unsure, you're probably not going to trust that data. But if the confidence score is high, and you see this validated in experimental data, I think this is when scientists are really getting interested, right? Scientists always want to see experimental data in my experience. Cool.
And to be honest, it is part of the, as we say, right, the test is in the pudding, right? So the experiment validation, I think, triumphs all predictions. And one, I think, important aspect here for any model is that the confidence is not a absolute fixed number, right? As we do experiments, the confidence score increases or decreases. And the second is it sort of guides you to determine in which areas you want to build more experiments to build higher confidence. So it's kind of a guiding principle, I would say, for any model generation and the experimentation you want to do.
Yeah. Can you maybe talk a little bit about how you generate this confidence score? It's based on experimental data, I'm assuming, right?
Yeah, absolutely. So we look at, so again, going back to the slide, if you have this distribution based on the distribution of the model, you'll do predictions, so prospective predictions, and then you test these predictions on experimental data, and that starts giving you a confidence score of whether these predictions, the predictions to the actual experimentation, how correlated they were. And that's how you backtrack, and based on this correlation, you kind of generate the confidence score.
Cool. Yeah. Earlier in the conversation, you mentioned that, like it's important that Edison has to integrate with this retrosynthesis tool, Cynthia. Can you maybe tell us a little bit about why you think that this is important and how this maybe differentiates?
Yeah, so as we looked at different models and we looked at ADMET data, fundamentally towards the end, we want to be able to make it. I think we kind of echoed this multiple times on this talk already. And so how do you ensure that the molecules that the model is generating right in the design phase has some synthesis feasibility estimation? And this is where fundamentally the problem was in most of the model prediction. Right? You started with predicting models, you started with predicting molecules, maybe they had good or bad ADME properties. And then after spending weeks of design, if you determine that these compounds cannot be made, that's a huge amount of wasted effort for a medicinal chemist. And so we determined that it is very important that we introduce synthesis feasibility very early in design. So step one is where you kind of take this into account. The other place where synthesis feasibility is important is the last step, which is the retrosynthesis design. So once you've distilled down your molecules from thousands of data points to a few hundred that you want to make, that's where you want to know what is the retrosynthesis pathway to be able to make these much faster and feasibly, either in your lab or you want to outsource it, but to be able to make them at the bench is critical. And so those are the two areas where we can bridge the gap with synthesis and truly take it from virtual to reality.
Awesome. Yeah, that's super, super cool. And yeah, so maybe can we talk a little bit about how you would then use that entire platform from like on a day-to-day business for like a medicinal chemist? Is it like, I'm imagining, I've had maybe a set of hits or something like that where I have like thousands of compounds in my library, maybe hundreds of thousands of compounds. Then I maybe do like, property predictions for this large set of compounds. And then from that set, I choose maybe a subset of compounds that I then predict the properties for. And from those, then I would put few of them in that retrosynthesis tool to get me maybe the top 10 compounds that I can synthesize very rapidly. Is that how you would use it, or am I totally mistaken here?
Totally mistaken.
Yeah.
Absolutely right, Dennis. Absolutely right. That's exactly how, you would use Addison, is to be able to use the generative models for design, right, to exploring these chemical spaces, and then you take it to synthesis.
I wanted to chime in here briefly. I got a question in the chat that was saying, thank you for the Q&A, but wouldn't it be great to let Ashwini continue with her presentation? So I just want to apologize for the confusion. This is meant to be a Q&A style interview with Ashwini. We're all on the same page, so we're not preventing Ashwini from giving a talk. This is how it's meant to be. We should have said that up front. But...
Yeah, sorry about that. That was my fault.
No problem at all. I think, thank you, Dennis, for clarifying. I think we wanted to, one of the fundamental things I think we recognize is the webinars are usually, I know it's monotonous, one person talking to everybody else. But I think this kind of conversation really helps to keep a dialogue going through which we can kind of maybe find better insights and newer insights.
Yeah, absolutely. Absolutely. I also like to hear like multiple voices. So yeah, that's the format that was meant to be. And I apologize that I didn't introduce it because it's a new format for the Flash Talks. We haven't been doing this in the past. But yeah, Ashwini, maybe you can tell us a little bit about how Edison actually got started. I think that's a really, really interesting story. And yeah, maybe you can tell the audience a little bit more about this.
Yeah, this story is very close to my heart. And I'm going to keep this image a bit here. So if you guys haven't read, you may want to just read this quirky story of, designing on a napkin that we started together. Addison truly started with, two scientists coming together at a conference, at a coffee table, and discussing what are the challenges that they face today, despite of the multiple tools that they use in drug design. There's still fundamental challenges when it comes to finding, new molecules that are either they have freedom to operate in this space, as well as that have robust predictions. And so this is where we started off this discussion and kind of sat together and said, well, we understand the challenge. Now, how can we find a solution? And as all scientists, we're trained, we're born and trained to find a solution to a problem. And so that's where we started really thinking beyond the problem and identifying technologies, both in terms of 2D, 3D structure design, as well as generative models, and starting to guide the generative models with better data, with better prediction, and multiple rounds of experiments. I will say we were not successful the first time. And I would be surprised if anybody would be successful the first time. So it took multiple iterations for us to be able to train these models to determine that we were on the right path. Sometimes, there were times where we had to trash the models as well if they were not a good prediction. But that, to me, is a testament of a good scientist. You continue to iterate and you continue to sort of challenge the models through the process. But that was our idea to innovation, and that collaboration between that scientist is super, super important in order to bring this innovation to bear.
And you already mentioned that scientists at Merck use this pretty frequently, right?
Yeah, absolutely. So as we kind of rolled out Addison, and as we were developing, I think that is even more important. As we started developing these models, we always had the scientists tested, because one can stay in in their own bubble and box and kind of convince themselves they're the best models in the world. But it's the test with the actual medicinal chemists in a group that was really critical. So they kept testing these as beta testers. And I really appreciate they were guinea pigs, but they continued to beta test as model as we kept training them and they got better. And that was really important for them to be able to adopt these models at the end and build confidence in the process.
Oh, yeah. So what is actually next? What do you think is like, this is a really cool model, but what is next on the horizon for Edison? What are your your visions for how the tool is going to develop over time?
Yeah, so I'm gonna go a little bit off track here and kind of share a fun image before we go into Addison. We talked about a lot of promises, right? Generative models, it's excellent, but there is a reality check as well, right? We have to be cautious about how we use these models. And I tried a little experiment. So I asked the model, a generative model, to recreate an image of, this this point in time, right, where Daniel and I met together and we ideated this whole Addison on a napkin. And this is what I got. I think it did a great job kind of getting two scientists together, female scientist, a male scientist. I had asked it to be German, but I think it did a good job at it did. And I think, so while, I'm highly impressed with the capabilities of the Gen AI to build such an image, if you look closely at the image, it still has fundamental problem in recognizing, even designing fingers. As you can see, the male scientist has 5 or 6 fingers, and some of the fingers are larger, smaller, the way it folds. So this has been and this is very important, that to identify that these minutiae and these details of these models are still not perfect. So while at high level, it may seem like a rosy picture, but we haven't solved it all. There's still a lot of work to be done in terms of training good data, in terms of continuing to reiterate these models, fine-tune these models so that they can, build reliable prediction. So I thought this was something fun to see what generative AI can do.
Yeah, definitely a nice test case here for image generation, and that's certainly something that may be even less complicated than medicinal chemistry. So yeah, maybe before we open it to the audience, one last question that I wanted to ask you is, where else do you see generative AI impacting medicinal chemistry as a field in general?
Yeah, so as a field in general, I think there are many areas. We kind of delved into drug discovery, particularly specifically today. And this is where, by the way, just to answer your previous question, this is where we're driving Addison in the future as well, is to be able to expand the capabilities, or I would say the modalities beyond small molecule into large molecules and new modalities, including whether it's mRNA, antibody, and others, PROTACs, right? That's where I think AI has a huge, I would say, opportunity to develop. It's really not there yet. I think it's very early in its inception. A lot of data is going to be needed, especially given that the proteins have so much thermodynamics and kinetics around it. We really have to generate a lot of data to be able to do better predictions. And the other area, of course, just saying in the medchem realm, not going to downstream, is also target identification, validation, network biology. And then on the preclinical side, in terms of getting the organ on the chip and being able to generate some of these models where we can try to be as in silico as possible before we go into humans or we go into animals for testing.
Thank you. So
Awesome,
I
yeah.
actually saw, I think there was a point that you didn't make here. I saw in the chat, there was a question, how can biotechs who don't have, access to this big pharma data leverage generative AI to do these, predicted properties and so on, do design? I think you haven't mentioned yet that this is actually a commercial tool that anybody can license this. Is that right? So like if you're at a small biotech, you can use this Merck KGaA data essentially.
That's right. And that's exactly the answer. Thank you, Dennis. But that's absolutely right. So and that's the question we asked too, right? If you're a small company or mid-sized small company, how do you get access to such a data? And that was fundamentally why we put the healthcare data, which is our own pharma data, into Addison. And since it's a commercial, an ability to make it commercial so that all the other scientists, whether small, mid-sized companies, would have access to it.
Yeah,
In
sounds
addition to that, we're also able to add data from other companies. So let's say a biotech has its own small specific, let's call it therapeutic area or a data that they've generated. We can also train the model on that specifically for them as well. So I think that kind of brings a holistic view, both in the therapeutic area of interest to them, as well as the larger data set that they get from MilliporeSigma or KGaA.
Interesting. Yeah, it's a nice way to democratize access to generative AI, I guess, to to the wider community if you don't have a massive library like Pfizer or Merck. I'm sure many companies have internal tools. Well, thank you so much for sharing this and being honest about the problems and challenges with this. I guess this is a much more complicated problem than honestly I expected going into this talk. I think you're sharing the notion of this confidence score. I think that's really helpful to be able to know if does the machine even think its own predictions are helpful or not. I think there's been a lot of hype about, these computers they're just going to take a molecule all the way to a clinical candidate right out of the box. It's clear that that's not how this actually works. But still, this definitely looks useful when you think about common problems that we talk about all the time. How do you search an HTS library for a subset of compounds that match a structure that you're looking for? How do you clean or curate an HTS library when you have millions of compounds? I mean, this is a great way to, scan millions of compounds at a time for permeability, or these other properties that you talked about. And I had a question about, can you do like similarity scoring, right? One of the big problems in drug discovery has always been, how do you compare one library to another? You want to go to 3 different CROs. How do you know if WuXi's library is better than, enamines or so on?
Yeah, I mean, it's hard to tell which one is better than the other, right? When it comes to chemical space exploration, whether you're trying to do, fragment, linking, scaffold hopping, or even decoration, the idea here would be to take both the approaches, right? Be able to explore all the spaces holistically. And that's the one thing we kind of made available in Addison is we have libraries from WuXi, Enamine, and Sigma-Aldrich put together in one large space, let's call it that way, which is about 10 to the power 20. And this was fundamentally important because there shouldn't be, I think a medicinal chemist shouldn't have to have a preference over one or the other, should be able to explore the largest chemical space possible. And the other is looking at the unknown chemical space with the reinvent models that are there in the back end.
So Dennis, thank you, Ashwini. So Dennis, there are a lot of questions in the chat. Maybe you and I can take turns because there's so many and read them one at a time.
Yeah, absolutely. That'd be awesome.
I'm gonna pick the shortest one first, and then you can take one. How about that?
All right, yeah. Go.
All right. So one question that I had as well was from an anonymous attendee is just how much of the proprietary Merck KGaA data was used in the model training?
Yeah, this slide speaks 1,000 words here. So we had about 30 years of experimental data that we distilled down. We have thousands of ADME endpoints and tens of models in the back end. So it's not one answer to all models. Some of the models could be trained on small amount of data, so it could range from even 3,000 data points, while some others actually need much larger data sets. So where we have smaller models, a smaller data set, we use regression or techniques of transfer learning to be able to embellish these data sets or these models. Where we have larger data sets, like thousands of data points, and one of them we have about 60,000 data points. There, I think some of the advanced deep learning models help. So it's a broad range depending on what assay endpoint you're looking for, or what I would say property endpoint that you're looking for.
Thank you.
Yeah. One question that was also in the audience that I had too. We talked a lot about property predictions, right? Is there a way to do potency predictions as well with Addison? So one of the questions in the audience was, how does Addison predict a molecule binding to the target? Like, could you maybe talk about maybe potency predictions a little bit?
Yeah, so we do have some binding assays in already, and that's where that uses, so the models use that data for binding. But in addition to the generative models, we also have docking. And so we are able to do the binding, get the binding score right off the bat with computational tools. And we extend that, or we take that binding score into account for and match it up with the assay data that we have.
Interesting. So if your target doesn't have any binding data yet, you'd have to rely on docking.
That's correct.
Okay.
Or then do the experiments, which I think is always the best way.
Okay, makes sense. So the purpose of this is mainly to pre-filter things before you put them in try to test them for IC50, for example. Just figure out what to make ahead of time. Okay. So a simple question here is, what does generative AI mean for inventorship and patentability? And that might not be an answerable question right now, but what are your thoughts on that?
Yeah, so patentability, I think is, and I kind of talked about this, the freedom to operate, right, in the patent infrastructure or molecule design is critical. One of the ways we're trying to address it, and this will be part of the future, some of the implementation you'll see in Addison this year, we are also incorporating the patent databases that you can find on Google Patents and others to validate the molecules that are generated from the generative AI. So it'll always be validated to see to the best of its capabilities, right? Based on the data set. Again, that's going to be crucial to have a really clean data where we've spent significant amount of time, even from patent database, is to generate this, clean data from patents. And then the models can be governed by this data to identify which chemical spaces has more freedom to operate, right? i.e., which can be more patentable as opposed to other.
Interesting. Actually, that's a use case that I hadn't thought about for AI, like determining whether you have freedom to operate. That's actually pretty cool. But I think the question that was being asked might be getting more to, let's say a generative AI designs this molecule, right? Or comes up with this molecule. I mean, is that molecule still patentable, or what's the current thinking there?
Oh, I see. So the current thinking, and again, this is based on multiple panels that we've been at in conferences. The general sentiment of the industry and the subject matter experts is, including the FDA, let's call it that way, and the regulators in the room, is to determine that, or is to attribute the IP to the scientists and the company that that is actually generating it, or the scientist that's generating it. AI is a tool to be able to do that, right? But at the end, it's the IP of the scientist to be who's kind of ideated the whole the molecule and the ideation behind it. So I would say that it is still the IP of the scientist, and I think patentability is hasn't been an issue so far. We've seen multiple molecules, like about 19 candidates that have come through that are preclinical trial at the moment that have been used, that have been generated using generative AI models, with the IP remaining with the parent group and the scientist.
Makes sense.
Does
Sounds
that
like it
answer your
relies.
question?
I think so, yes. It relies more on reduction to practice, whoever's reducing it to practice. I mean, there was a similar debate a long time ago about, Marcouche structures, right? And, if you patent a Marcouche structure, you claim the entire universe. And I think it came down to, well, it's whoever reduces it to practice and shows evidence of that. Okay, Dennis, do you have another one?
Yeah, maybe one here from Antran at Amgen. She's wondering if Addison will have different predictions depending on the stage of the program. Say, if you're in like a hit optimization, in a hit identification or in a lead optimization, stage of the program, will there be, stricter criteria if you're in a lead optimization, or, will Addison adapt, I guess, to the stage of, where the project is at the moment?
Yeah, really good question. I think fundamentally the answer is yes. It would depend on how you use the model and the tool. So if you're looking at when you're hit identification very early on, you can essentially loosen the parameters and try to look at the unconstrained space with unconstrained space, unconstrained molecule ideas. But when you go to optimization, we actually try to focus more on fine-tuning the molecule, either the scaffold or the decoration around it. And there we try to focus more on glowing twin and iterating and looking at the impact of the prediction based on every molecule that you iterate. So totally fundamentally different approaches to get predictions, reliable predictions from the model.
Thank you. One question from Jonathan Thielman is, do you make efforts to do blinded validation of the predictive power of these models? So for example, for the KGaA2 prediction, have you conducted studies or done retrospective analyses where the predictions are made for hundreds of compounds with unknown KGaA2 permeability and then actually collect the data empirically and see how well they match up?
Yeah, we do. So part of this on this slide, when we talk about retraining, that's exactly what we do. So we take brand new, we've been working on many different chemical spaces or therapeutic areas on this, and we do experiments along the same lines and then train the model to be able to retrospectively identify what the scores would be, and then also prospectively predict what the scores would be for the next set of molecules. We also published a paper around this on retrospective study. So this is where we really had the model predict some of these, and then we went back and saw, did it match with the experimental data, historic experimental data.
Maybe another question here is about the, I guess, quantity of data or compounds that the model was trained on. Like, can you maybe give people an idea of how many compounds have been, synthesized to better test the model?
Well, I don't have one number.
Yeah, probably large.
It's been, I mean, in the last 30 years, you can imagine the number of experiments, right, that have been conducted by the scientists. I think this is millions of data points by now that were experimentally done, but distilled down to a few, couple of thousands of data points, let's call it that way, right? Or low digit million data points. So we wanted to make sure that all that experimental data is actually leveraged, because it is extremely expensive. As much as I'm a proponent of doing experimentation, it is extremely expensive to be able to do that. So I think we've made sure that we leverage that experimental data, but in the future, and that helps us to determine the fewer set or the smaller set of experiments that we want to do moving forward.
Now that's actually a super interesting point about the cost of each experiment. Like, like KGaA assay probably is like in the maybe $1,000 range actually per experiment. So it's pretty expensive assay. I'm wondering, I guess that's why you're probably updating the data live with new compounds. Is that the case? Look, every compound that's synthesized at Merck and that's assayed, in any of the, I guess, ADME assays at Merck is added to the data set. Is that right?
Oh, absolutely. Every new data. So as we generate as we do new experiments, all of that data gets retrained to the model. Especially even the same molecules that may be tested differently. And if we have different results, then we optimize the models with the new data points.
Awesome. Yeah, thank you. That's super interesting. Dennis, you have another one?
Yes. So let's see. Patrick Krajewski asks, how do you want to build higher confidence scores for your predictions in Addison if your data set is fueled only from Merck's in-house medchem campaigns?
Yeah, that's a really good question. Well, to generate confidence score, the experimentation is going to be key. And that's going to be, and so this is where we're going to need hundreds of users to be able to use Addison, generate predictions, test it, and see whether this data, and then sort of bring that information back to retrain the model. So I think it is going to be important to expand the data set beyond healthcare, right? This was our starting point. Rather than going from just public data, which is, again, like I said, not really correlatable and not robust from the experimentation point of view, we wanted to start with robust data, but we definitely plan to expand the database. For example, we just signed an agreement with Accelera for their Koester database, which is another curated data set. And we're going to enhance our models with that data set. The other way to do that is, like I said, when companies, users start using it, some of their data model could be retrained on that data set together or in parallel. And that also allows to make the models better. So I think there's a lot of room to improve on this area. And happy to collaborate if you have an idea on that.
Yeah, actually, speaking of that, that reminds me of something we talked about one-on-one a month or so ago about, I think we take it for granted as scientists, like, okay, we just get the data, we throw it in the model. But I think you mentioned that like from a business development standpoint, even internally, it was incredibly difficult to set up the information security and the like contracts to enable this. I mean, can you talk a little bit more about in practice how you made this, this thing work?
Yeah, really good question, and sort of brings not so fun memories back. But on a more serious note, I think as much as collaboration is important, and that's where we started driving, one of the important aspects is to ensure that we have security around these models and the data that's coming from pharma. And it's quite important from two aspects. One, we do not want the traceability to the IP, which is super critical. This is the IP of the parent organization, and would be for anybody else. This is how we treat every user, every company. Their data security is of utmost importance to us. So we ensured when we started training these models, actually, we put some technical sort of models in place, or say technical constraints in place that sort of prevent from backtracking the data. The second thing was ensuring ISO certification. So this is the highest standard in software for information system management. And so before we rolled out Addison commercially, we ensured that it is ISO 27001 certified. So that really takes into account security in the cloud, information sharing and information management, data management across the platform.
Okay, looks like we are down to the last minute. So Dennis, Koester, do you have any final questions or thoughts?
Yeah, maybe one last question. That's a good question, I guess, to wrap it up. What are your top 3 tips for designing a software for medicinal chemists?
Top 3 tips.
Oh, we can do the top 1 tip if we only have a minute, right?
Well, top 3 tips would be to start with a clean data set. So spend some time there. Try multiple models with the data set and try to compare it to the experimental data, like, Dennis, we talked about this retrospective testing. And stay close to your data scientists need to be very, working in collaboration hand in hand every day with the medicinal chemist. That I think that knowledge sharing between the two is extremely, extremely important to ensure that the predictions out of the models are actually robust and have been tested by the scientists. So those are my top 3 tips.
Awesome. Yeah, thank you so much. And I guess, yeah, that almost wraps it up. We're at the top of the hour. I guess, Ashwini, you wanna say some closing comments before Dennis and I close it up?
Sure. This was amazing. I will say I've been, through the conversations, with both Dennis, I was going to say Dennis Square to be a bit nerdy. It's been amazing to kind of, the progress that I've seen of Drug Hunter throughout the years has been remarkable. So hats off to your team. As I said earlier, I'm going to close with that. It's been a remarkable amount of knowledge that helps medicinal chemists, drug discovery scientists across various levels in the industry to stay on top of this. And by the way, we always look at the molecule of the month and use it for testing Addison. So a lot of our application scientists always keep an eye on that and kind of try to look at the predictions and so on and so forth. So it's been extremely helpful to us. So thank you for that, and thank you for the invitation. to talk on this important topic with the audience.
Thank you for being here. It was really phenomenal. Thank you for sharing your thoughts. I think it was really, really great. And I personally learned a lot. So yeah, thank you for an engaging talk. Thank you, Dennis, also for being here and helping with the audience questions and everything. I couldn't have done it by myself. Thank you to you in the audience. It was a great engaging discussions. I know we had many more questions than we could answer. So stay tuned, maybe for an update on our webpage on the Q&A. And yeah, again, we look forward to seeing you at the next event and see you then. Have a great rest of your week. Bye for now.
Thank you, everybody. Thank you, Ashwini.
Thank you.
Great rest of your week.